In this video, we'll explore the role of DNA as the carrier of genetic information. What do we know so far? Genes function as units, and genetic experiments suggest that chromosomes are linear arrays of genes. Additionally, chromosomes contain two types of biomolecules, proteins and DNA. What does this information allow us to infer? We can infer that the molecule that contains hereditary information should be located in chromosomes. Since proteins are unlikely to contain hereditary information, DNA must be the molecular support of heredity. Although it may seem simple, it took 25 years for the scientific community to accept this conclusion. 3 significant scientific discoveries were crucial in determining the role of DNA. The first was the identification of a transforming principle in pneumococcus, a type of bacteria that can infect humans and mice and causes pneumonia. The second was demonstrating that DNA is the transforming principle in pneumococcus. The third was demonstrating the role of DNA in bacteriophages. Let's focus on identifying the transforming principle in pneumococcus. There are different pathogenic strains of pneumococcus, namely types 1, 2, and 3. Which are distinguished by the composition of their polysaccharide capsule. This capsule is crucial for pneumococcus's pathogenicity. When grown on solid medium in a petri dish, pathogenic wild-type strains form smooth colonies, denoted as PNS, while non-pathogenic mutant strains have a deficiency in their capsule structure and form rough colonies, denoted as PNR. The PNR strain is easily recognized and cleared by the immune system. Mutations can convert pathogenic strains into non-pathogenic strains. The frequency of mutations that are responsible for conversion of PNS to PNR is about 10 to the minus 6 to 10 to the minus 7. This means that out of every 1 million to 10 million bacteria, 1 might undergo this mutation. The frequency of reversion, conversion of PNR back to PNS, is about 10 to the minus 9. In 1928, Frederick Griffith conducted experiments to test pneumococcus's pathogenicity. His approach involved infecting mice with live pathogenic pneumococcus, live non-pathogenic pneumococcus, or heat-killed pathogenic pneumococcus, and comparing the results. In the first set of experiments, a wild-type live pneumococcus strain was injected into mice. These mice developed pneumonia and died, which demonstrated the pathogenic nature of PNS. In contrast, when a mutant live pneumococcus strain was injected into mice, they survived and did not develop pneumonia, which demonstrates the non-pathogenic nature of PNR. The next set of experiments involved injecting mice with heat-killed wild-type pneumococcus. These mice did not develop pneumonia, which confirmed the elimination of PNS's pathogenicity through heat. However, an unexpected result occurred when mutant live PNR and heat-killed PNS were mixed. This combination, when injected into mice, caused pneumonia. This suggested that non-pathogenic PNR became pathogenic when mixed with heat-killed pneumococcus. Bacteria that were isolated from the dead mice formed smooth colonies and thus exhibited the pathogenic phenotype. Griffith hypothesized that the conversion into a pathogenic strain, termed transformation, was the result of a compound being transferred from the heat-killed PNS to the live PNR. He called this compound the transforming principle. Is there any other explanation for these results? Griffith considered the possibility that the newly found pathogenicity of PNR could be due to the reversion of the PNR to the PNS phenotype. To test this theory, heat-killed pathogenic PNS and live non-pathogenic PNR were grown separately on solid media. Heat-killed PNS alone did not form colonies, while live PNR formed mostly rough colonies, with some smooth colonies at a frequency close to 10 to the minus 9. However, when live PNR was mixed with heat-killed PNS, smooth colonies formed at a much higher frequency, closer to 10 to the minus 2. This indicated that the observed transformation could not be explained simply by reversion, but rather by a distinct process. This demonstrated that reversion and transformation are two different processes that occur at two different frequencies. What is the chemical nature of the transforming principle? Scientists Avery, McLeod, and McCarty provided the answer in 1944. Their experimental approach was straightforward. They started with live nonpathogenic PNR bacteria and plated it on solid medium with extracts of pathogenic PNS. The extracts were either left untreated or treated with enzymes to selectively remove carbohydrates, proteins, RNA, or DNA. The bacteria were allowed to grow on solid medium to determine whether transformation took place. If there was no transformation, smooth colonies would appear at a frequency of about 10 to the minus 9. If there was transformation, colonies with a frequency close to 1% or 10 to the minus 2 would be observed. This table covers the key components of the experiment: the state of PNR, dead or alive, PNS extract, which enzyme was used, hydrolyzed compound, which components of the PNS extract had been removed, and transformation, whether or not PNR transformation took place. Mixing heat-killed PNR with untreated PNS extract resulted in no transformation, as expected. When live PNR was combined with untreated PNS extract, transformation took place. If the extract was treated with the enzyme protease to eliminate proteins, transformation still took place. If the extract was treated with the glycosidase enzyme to eliminate carbohydrates, there was still transformation. This means that neither carbohydrates or proteins are involved in the process of transformation. If treated with the RNase enzyme to eliminate all RNA, transformation still took place. However, when treated with the DNase enzyme to eliminate DNA, no transformation occurred. This indicates that proteins, carbohydrates, and RNA are not necessary for transformation, as their elimination still allows for transformation. Therefore, DNA was shown to be the transforming principle in pneumococcus. Avery, McLeod, and McCarty questioned whether DNA alone was responsible for the observed transformation in these experiments. Instead of using PNS extract treated with DNase, purified PNS DNA without any contamination by proteins, carbohydrates, and RNA was used. Purified DNA also resulted in the transformation of PNR into pathogenic pneumococcus. The second question addressed whether the transformation is hereditary. To determine this, they considered whether transformation is the process by which PNR bacteria are converted into pathogenic smooth colonies. Since all the bacteria in a smooth colony originate from a single transformed PNR bacterium, could this transformed phenotype be passed on to all subsequent generations? The answer is yes, otherwise a smooth colony could not form. The transformed phenotype is indeed hereditary, as it is passed on to all generations of bacteria within a single smooth colony. The third question examined whether the transformation is specific. There are 3 types of pathogenic pneumococcus, types 1, 2, and 3, and PNR can be transformed by the DNA of a specific type. For example, type 1 DNA produces type 1 pneumococcus. This indicates that the transformation is indeed specific. The fourth question considered whether the transformation is restricted to one phenotype, specifically in terms of pathogenicity or the ability to form smooth colonies. The experiments showed that traits other than pathogenicity could be acquired through transformation, such as resistance to antibiotics, metabolism of specific sugars, and the ability to synthesize specific proteins. Therefore, the transformation is not restricted to one phenotype. In conclusion, these experiments conducted with pneumococcus demonstrated that DNA is the physical support of heredity. DNA also directs the expression of a variety of heritable traits, suggesting that genes are made out of DNA. However, the impact of these experiments were initially limited because the scientific community was hesitant to accept that a molecule as simple as DNA could direct the synthesis of complex proteins.